Good morning, students. Today we are discussed about a new plant hormone, cytokinins. The discovery, identification, and property of cytokinins. A great many substances were tested in an effort to initiate and sustain the proliferation of normal stem tissues in culture. Materials ranging from yeast extract to tomato juice were found to have a positive effect, at least with some tissues. However, culture growth was stimulated most dramatically when the liquid endosperm of coconut was added to the culture medium. Philip White's nutrient medium, supplemented with an auxin and 10 to 20% coconut milk, was found to support the continued cell division of mature differentiated cells from a wide variety of tissues and species, leading to the formation of callus tissues. This finding indicated that coconut milk contains substances that stimulate mature cells to enter and remain in the cell division cycle. Eventually, coconut milk was shown to contain the cytokinin zeatin, but this finding was not obtained until several years after the discovery of the cytokinins. The first cytokinin to be discovered was the synthetic analog kinetin. Kinetin was discovered as a breakdown product of DNA. In the 1940 and 1950, Fouquet Cook and co-workers at the University of Wisconsin tested many substances for their ability to initiate and sustain the proliferation of cultured tobacco with tissue. They had observed that the nucleic acid base adenine had a slight promotive effect. So they tested the possibility that nucleic acids would stimulate division in this tissue. Surprisingly, aged or autoclaved herring sperm DNA had a powerful cell division promoting effect. After much work, a small molecule was identified from the autoclaved DNA and named kinetin. It was shown to be an adenine derivative, 6-furfuryl aminopurine. In the presence of an auxin, kinetin would stimulate tobacco pith parenchyma tissue to proliferate in culture. No kinetin induced cell division occur without auxin in the culture medium. Kinetin is not naturally occurring plant growth regulator and it does not occur as a base in the DNA of any species. It is byproduct of the heat induced degradation of DNA in which deoxyribose sugar of adenosine is converted to a furfuryl ring and shifted from the 9th position to the 6th position on the adenine ring. The discovery of kinetin was important because it demonstrated that cell division could be induced by a simple chemical substances of greater importance. The discovery of kinetin suggested that naturally occurring molecules with Similar to that, kinetin regulates cell division activity within the plant. This hypothesis proved to be correct. Zeatin was the first natural cytokinin discovered. Several years after the discovery of kinetin, extracts of the immature endosperm of maize were found to contain a substances that had the same biological effect as kinetin. These substances stimulate mature plant cells to divide when added to a culture medium along with an auxin. Lidham isolated the molecule responsible for this activity and identified it as trans 6 methyl butylenyl aminopurine, which he called zeatin. The molecular structure of zeatin is similar to that of kinetin. Both molecules are adenine derivatives. Although they have different side chains in both cases, the side chain is linked to the nitrogen attached to C6 of adenine. Because the side chain of zeatin has a double bond, it can exist in either the cis or trans configuration. In higher plants, zeatin occurs in both the cis and the trans configurations, and these forms can be interconverted by an enzyme known as zeatin isomerase, which is found in some but not all plants. Although the trans form of zeatin is much more active in biological assays, it is likely that the cis form also play important role in some plant species. For example, in Arabidopsis, the trans form predominates, but in maize and rice, most zeatin is present in the cis form. Furthermore, trans zeatin can bind to and activate cytokinin receptor from both Arabidopsis and maize, but the cis form is much more active with the cytokinin receptor from maize. Since its discovery in immature maize endosperm, zeatin has been found in many plants and in some bacteria. It is generally the most prevalent active cytokinin in higher plants, but other substituted aminopurines that are active as cytokinins have been isolated from many plant and bacterial species. These aminopurines differ from zeatin in the nature of the side chain attached to the sixth position of nitrogen, or we can say 9-6 position. These cytokinin can be present in the plant as riboside, in which a ribose sugar is attached to the ninth nitrogen of the purine ring. Ribotides, in which the ribose sugar moiety contains a phosphate group. Glycosides, in which a sugar molecule is attached to the 3, 7 or 9 nitrogen of the purine ring, or to the oxygen of dihydroxyatine, or in other conjugated forms. Thank you, students.